afternoon, everyone. I'm Kristen Kokeman, President and CEO of the ALS Association Greater New York Chapter. Thank you so much for joining us for our Education and Resource Summit and today's session, Genetic Causations in ALS, Simplifying the Complex, presented by Dr. Matt Harms and Elizabeth Harrington. Dr. Matt Harms is an Associate Professor of Neurology at Columbia University Medical Center. He sees patients in the ALS Association's Certified Treatment Center of Excellence at the Eleanor and Lou Gehrig ALS Center and directs Columbia's Precision Medicine Initiative in ALS. In addition, his laboratory straddles the Motor Neuron Center and the Institute for Genomic Medicine with a focus on amyotrophic lateral sclerosis and other neurological disorders. Elizabeth Harrington, or Ellie, is an ABGC board-certified genetic counselor and lecturer in the Department of Neurology at Columbia University, where she provides genetic counseling expertise in neuromuscular, neurodegenerative, and motor neuron diseases. Ms. Harrington received her graduate degree in human genetics and genetic counseling from the Stanford University School of Medicine. More information on both speakers can be found in the Speaker Center on the Summit platform. Today's presentation will run for approximately 1 hour and will include a question and answer session. Please use the chat function to send in your questions and they will be presented to Ellie and Dr. Harms. If we are unable to get to your question, we will respond offline, and there will be another opportunity on Friday during our closing session to ask follow-up questions. I'll now turn the discussion over to Ellie and Dr. Harms to get us started.
All right, great. Thank you everyone for coming. Can everyone see my presenter view? All right, great. So today I'm going to attempt over the next 20 or 25 minutes to simplify the very complex world of genetic causation in ALS and what we know as of today. To begin with though, I do want to point out that the timeline of ALS genetic discoveries has been quite robust. You can see that the first gene was discovered in 1993, and now we're up getting close now to 3 dozen. And I would like to thank ALSA for giving me the opportunity to speak about this because more than $10 million, much of that derived from the Ice Bucket Challenge several years ago, has gone to fund much of the work that we'll be discussing and hearing about today, especially in these most recent years. But in order to just understand the gene discovery timeline and what that means for your journey with ALS, I want to go all the way back to the human genome. So the human genome itself is 6 billion base pairs long. That is huge. By comparison, the coronavirus only has 30,000 base pairs. The human genome is made out of DNA, and all of that is packed into the nucleus of most of our cells. Cells will then use these different parts of the DNA as code for the proteins that they need in order to function. If you look across the genome, you'll identify about 19,000 individual genes. Coronavirus, for comparison, only has 29. But those 19,000 genes are actually used in very creative ways. So they actually manufacture around 80,000 different proteins. You can think of proteins for the rest of this talk as the building blocks of the machinery inside of your cells. Each protein that gets manufactured from a gene is a widget or a building block that allows the cell machinery to do what's necessary to be a good liver cell or a good neuron or a good muscle fiber. The proper protein function is key to our health. And I want to talk a little bit about how genes get turned into protein and how that goes wrong in mutations. But before doing that, I want to point out that of those 19,000 genes in our genome, around 6,000 of them are known already to cause human disease. So most of our genes are quite important for health. So how do individual genes make individual specific proteins? Well, the central dogma is presented there on the right. And basically it says that you start off with DNA, you copy it into a similar molecule called RNA, and then that RNA serves as the basis for manufacturing the protein. What does that look like in detail? So here is an array of DNA, one chromosome, for the gene CHOMP and linker. These are 2 different genes arrayed on the same chromosome. Because of the way our bodies come together, because of fertilization, we get 1 copy of the chromosome from mom and 1 copy of the chromosome from dad. And for most genes, both chromosomes get utilized when it's time to make the protein. So the first step is cellular machinery comes in, and it uses a process called transcription to copy each of the genes from each of the chromosomes. So you get now RNA for the CHOMP gene, one from each of the parents. Then a process called translation takes place, where another set of cell machinery goes along the RNA and transcribes it and translates it into amino acids, a code that will tell the protein what it's supposed to do. And because you have two normal copies one from each parent, that happens twice. So you'll end up with two normal amino acid chains that are going to go on to fold and become a protein. How does that happen? Well, each of these amino acids knows which other amino acid to stick to and to bind. And so it actually forms what we call secondary structure. So it starts to fold up according to the amino acid structure. And so it goes something like this. It first coils up a little bit based on that structure, and then eventually goes on to form its very final structure. In this case, something that looks a little bit like a Pac-Man, which is appropriate because I've humorously titled this the CHOMP protein. And the shape of most proteins gives you a clue as to what it's supposed to do. So in this particular case, just hypothetically, the CHOMP protein is supposed to find all of the purple rods and cleave them in half so that now you have two smaller purple rods, and the two smaller purple rods can go on to do what it is that they're necessary to do. Maybe build something like a highway that allows transport to take place inside of the cell. So that's the CHOMP protein. There are other very different proteins that come from very different genes. So the linker gene here, the green one, also undergoes transcription and then undergoes translation, and it will form a protein that looks very different, has a very different shape, one that's well suited to its linker moniker. So in this particular case, this particular gene, protein, is designed to bring the blue protein and the red protein into close proximity so that they can communicate with one another. Instead of floating around in the cell, they're right next to each other and can talk to one another. And so by this way, the body creates all of the building blocks that are necessary for a cell to function well. So how do genetic mutations interrupt this process? Well, shown here in the center for both the CHOMP and the linker are the normal routes for the normal copy. But let's introduce a mutation, which is just a disruption of the DNA. It's maybe been swapped out for a different base pair, or it's missing a little chunk. And so I've illustrated that by this little black dot on the mom's copy. Now, when the cell tries to do transcription of the mother's chromosome, you'll end up with an abnormal mutated RNA. And then as the translation takes place, you're likely to end up with a protein that no longer has the normal structure or function and looks something more like this. So it's still kind of a Pac-Man shape, but the mouth is way too open. And in fact, when you then try to have it cleave that particular rod, it doesn't happen. And so rather than generating lots of little small purple rods, you end up with accumulation of all of the big purple rods that needed to be cleaved. And that, of course, can go on to cause problems. Now, if we introduce a mutation into the linker gene on the father's copy, now when it undergoes transcription, it will end up with a mutated RNA, which will then give rise to a funky shaped linker protein, which is unlikely to work as well as the normal one. And so now these two molecules try to come in close proximity, but it's too shallow and the blue one drifts away, and now those two proteins can't communicate with each other. So that's the essentials. That's how mutations that are inherited give rise to messed up protein function, essentially. But there are several different ways that can end up working out inside of a cell. So many mutations are what we call loss of function mutations. Loss of function, meaning an essential thing that it's supposed to do just doesn't occur. So I've illustrated the normal situation here. You've got the CHOMP protein from the dad, the CHOMP protein from the mom, both of which are looking just fine. And they're trained and evolved to basically cleave the big rods into the small rods, the purple ones. So now, one of the ways a mutation can be a problem is that the mother's copy, if it were missing or severely mutated so that it didn't even get formed, you would have half as much of the CHOMP protein. And as you might expect, half of the rods would get cleaved and the other would be left standing. And so you would end up with too many long rods and too few short rods, and that could give rise to a faulty highway inside of your cell. You could also imagine that the mutation, like in the example I showed you on the previous slide, the mother's mutation could lead to a malformed and defective CHOMP protein. So now, even though it's there and you can see it there, it doesn't effectively cleave the rods either. And so you'd end up with a very similar situation. Too many long rods and not enough of the small rods. Many proteins actually pair together. So you end up with two copies of the protein working together in unison. And if that's the case, if you now introduce some messed up copies, they can actually stick to and impede the function of the remaining normal protein. So now you've got the normal CHOMP, but it's bound to and stuck by the abnormal CHOMP protein. And so now you'll actually get no cleavage at all, none of the small little cleavage products, and way, way too many of the large ones. And then in some cases, you don't even manufacture the protein at all, and so there's also no cleavage. Here are some of the ALS genes that are in these categories. There aren't very many. Most of the genes that cause ALS are what we call gain of function, but there are a few that I've illustrated here. Gene therapies for loss of function mutations have to do a couple of things. So as they're being developed, they have to boost the good copy so that you now have enough of the protein to do the job. And it has to block the bad copy so that you don't end up interfering with the function of the good copy. And if it's missing altogether, of course, you have to resupply or give a good copy of the gene in order to repair the damage. Now let's move on to the gain of function mutations. Same type of diagram, dad's orange CHOMP proteins, mom's red. And now I want you to imagine that basically they're still supposed to cleave the purple rods into small rods, but they're supposed to leave the blue ones alone, and they've evolved to be able to do that. Now, a gain of function mutation occurs when you have too many of the CHOMP protein, or the CHOMP protein that's produced is overactive, or it can't tell the difference anymore between the purple rods and the blue rods. And so when you've got too many or they're too active, they go willy-nilly, and they basically chop up indiscriminately both the purple and the blue. So now you don't have the long blue rods you needed, and on top of that, they can continue chomping, so you end up with way too many small fragments instead of intermediate-sized chunks. And then you can't build anything with those. Another thing that can happen is you can inappropriately turn the gene on in a place and time when it's not supposed to be turned on. So it can be activated in the wrong cell, it can be activated in the wrong part of the cell, and it can be activated at the wrong time. So perhaps this is a gene that's supposed to be on during development, but then once you're an adult, it's supposed to turn off. And now all of a sudden, it's back on when it's not supposed to. And so you end up having overactive cleavage of the blue rods, the purple rods, in a time and fashion that it's not supposed to occur. So that happens in ALS, but by far and away, the most common gain of function mutation mechanism that we encounter in ALS comes about with misfolded or abnormal proteins that aren't even supposed to be there. And in these circumstances, we don't think that it has much to do with what the gene was supposed to be doing, but by the acquisition of a new problem coming from the misfolded protein or from the generation of new protein. So what happens when proteins misfold is they become sticky. The new proteins that get manufactured are also sticky, and so they tend to clump up and form like concrete hard globs inside the cell. Sometimes these hard globs aren't made of just the bad misfolded protein, but they actually accumulate and suck in the normal protein that you needed and makes an even bigger problem. Similarly, those new proteins that aren't supposed to be there can also cluster and clump up and cause problems. The examples are some of the most common mutations that we find in ALS. SOD1, the C9orf72 gene, the gene called TARDBP, and the gene called FUS are all examples where this abnormal clumping of misfolded or new proteins takes place. Gene therapies that might go after these have to do two things also. They have to be able to block the bad copy so that the clumping doesn't occur, or tackle the clumping itself and keep that from occurring in order to keep cells healthy. Let me just explain this one other way by saying that mutations are not all equal. So this is that CHOMP protein again, and here's the normal circumstance, the normal RNA without the mutation and the normal protein. A mutation introduced here in the middle might partially affect the Pac-Man and make it less efficient at cleaving the purple rods, whereas a mutation located here at the very beginning might completely destroy the ability for cleavage to take place. And so you might end up with cleavage that looks like this: normal cleavage, half the amount of cleavage, and then absolutely no cleavage at all. So if this is a clump-prone protein, in the normal circumstance, there will be no clumps and happy neurons. In the less efficient circumstance, you'll have some clumps and neurons may be able to cope for a while. And then in the completely destroyed situation, you'll get many clumps, which are undoubtedly going to lead to dying neurons. So what would this look like kind of in a human being? Well, in a human with a normal CHOMP protein, there would be no disease. In an individual with a less efficient mutation, you might have some individuals in the family escape developing ALS altogether. There might be later onset of ALS because it takes time for the clumps to accumulate, and there might be slower disease progression. Whereas if you have a really bad mutation that destroys the protein function, you might end up with no one in the family who has the mutation escaping, early onset in terms of age, and then a faster disease progression. And finally, one other way to look at this is by this balancing scale. This is the inside of a neuron, and there are going to be insults that threaten the cell and compensations that try to keep it from going south. So a big bad mutation, like the completely defective version that I just showed you, can insult the cell, and the body will try as many compensations as it has. But eventually, the clumps will be too bad, and neurons will die. There are other mutations, even within the same gene, that aren't quite as bad. One example would be the SOD1 I114T mutation. And for a while, the body is able to compensate, and in some people, it's able to compensate for the whole lifetime. But add in one other genetic factor, like being male and having an X and a Y chromosome instead of two X chromosomes, and that can be enough to kind of tip the balance and mean ALS would occur. And then there are mutations that barely even register and should be really considered risk factor mutations. So these can be any number of genes, even some environmental factors that all accumulate as insults. And then your body is able to compensate for most of life. And in most individuals, compensates for your entire lifespan. So ALS never develops. And then in just the right individual, another genetic abnormality or even an environmental exposure might land on the insult column and tip the balance, leading to ALS. So ALS is a complex genetic disorder, and there's a spectrum of things happening. There are individuals who have really bad mutations that are virtually inescapable, and ALS is bound to ensue if you live long enough. There are individuals who have more mild mutations where it's likely that you'll get ALS, but some people escape. Another set of individuals where there are multiple mutations working together to trigger ALS, and then some individuals where undoubtedly there are many. We call that first group monogenic ALS, and it can occur either in families or sporadic. We call the group with a few mutations oligogenic, and these individuals usually appear to be sporadic. And the individuals who have many, we call polygenic, and they also appear to be sporadic. Our current ability to discover genes is well established on this end of the spectrum. We're in the process of doing it here in the middle. And in the future, with more samples and other technologies, we'll be able to look and identify these polygenic examples. So let's go back to the discovery gene timeline. So here's basically all of the genes that have been discovered as of a couple of weeks ago. So you can see that there are quite a few of them. And you may be asking yourself, how common are these and how likely is it to be found? So here's a study that I did back in 2015, where we took 42 people with a family history of ALS, and we sequenced the 17 genes that were known at that time. We found an explanation in just over half of them, the vast majority being from the C9orf72 gene, followed by the SOD1 gene. The sequencing technologies were just cheap enough at that time for us to start to look in sporadic patients, people who had no family history. And we looked in 340, and we found that 1 in 10 of them had the exact same mutations we were finding over in the familial category. I've recently carried out a new analysis of 10 times as many individuals, now with the full 32 genes. And these numbers roughly sort out still. About 1 in 10 individuals with sporadic disease and around 60% of familial disease, we can identify mutations. I'd like to point out that the gene burden that I've just showed you applies mostly to European populations, and it will differ depending on your geoancestry. C9orf72 is quite rare in non-European populations, whereas SOD1 and the FUS gene and even TBK1 can be more common in Asian, Indian, and African ancestry. So it does matter what your racial or ethnic background is in terms of what gene may be the most likely to be identified. So I just said that 1 in 10 patients with apparently sporadic ALS, no family history, turn out to have these gene mutations. How does that occur? Well, one possibility is that there's missing family history. Some individuals are adopted, sometimes without even knowing it. Other times, you don't know the full family history because an early death occurred before symptoms would have come on, and it's possible that a parent would have gone on to develop ALS. You can also have misleading family histories. Some family members might be diagnosed with a neurodegenerative problem that's not recognized to be part of ALS, like dementia, or a very atypical case of Parkinson's disease. Sometimes the diagnosis is missed in a family member. So someone is diagnosed as having a muscle disease, when in fact it was actually ALS all along. It just didn't get diagnosed properly. And then finally, on rare occasion, some people have different biological parents than they assume they have. And if they knew the true family history, you would see ALS on one of the sides. Another possibility is that when the egg is being created or the sperm is being created, a spontaneous mutation occurs in one of those genes. This probably happens in ALS, but we think it's pretty rare. In the circumstances where we've been able to look, it's just extraordinarily rare. But the most common reason is going to be lower penetrance or a smaller effect of the mutation itself. Kind of like that I114T mutation I showed you that was the small weight. Most of the mutations that we're dealing with in ALS are insufficient all by themselves to guarantee developing the disease. And so some generations get skipped. Maybe they have a genetic factor that protects them. Or maybe the individual who develops the disease has a second genetic factor that makes them more likely to develop disease. So back to the relative gene contributions, you'll notice that a large number of individuals don't have identifiable genes at this point. So how many more genes are there? I've estimated that there are about 100 at least. And we need to find them. And so how are we going to do that? Well, the traditional route to gene hunting was to find a big family with ALS, find the region of a chromosome that they shared, and then sequence that until you found the mutation. That was a 5 to 10 year process, but it was successful. We found SOD1, the FUS gene, and a couple of others. But we ran out of large families to help us identify these new genes. Now, don't hear me wrong. We don't want large families with ALS, but we've studied all of the ones that currently exist. So now you can take any family with ALS where there are at least 2 affected individuals. And now instead of doing all of that labor-intensive work, you just sequence the whole genome over the course of 1 week. Actually, it's kind of more like 2 days now. And then you look for families across multiple centers that have the same gene mutated. We use this, not me personally, but the field to find VCP, ubiquilin-2, and profilin-1. And now we're actually doing what we call next generation hunting because we don't have many of those small families left either. So in this form, you take thousands of people with or without ALS, you sequence the genomes of everyone, and you look for the regions that have more mutations in ALS patients than in the controls. So here you can see on the right, there are far more lightning bolts, which I've represented as bad mutations on the cases side, the ALS side. And that allows you to statistically identify that this region of the genome is causative for ALS. We were able to be the first to identify TBK1 and NEK1 as causative genes using this technique, but others, as listed here at the bottom, have also been found using this technology. And it's the way we'll be doing it in the future. So if you now stratify the gene discovery timeline based on those methods, the blue is the old laborious method. The green are the genes that were discovered using exome sequencing in small families. And now we're in this era of burden testing in red, finding new genes on average once every 3 months. I think you'll appreciate that this looks a little bit like a COVID surge chart. And while we want the COVID surge to come down and go back to zero, of course, we want the ALS cases to just, or the ALS genes to just keep skyrocketing as we discover more and more genes. And why do we want to discover more genes? Primarily because they're falling into categories of different functions within cells that are important to cell health. And we can now go back and try to design drugs that target RNA metabolism, target protein homeostasis, or axonal transport that may be useful across many patients with ALS. The goal would be that when we look at the clinic of the future, we would take every ALS patient that comes in and do whole genome sequencing. This would allow us to, by genetics, stratify them into the different groups based on their driver mutations. Maybe some of these are neuroinflammation, cellular transport, protein recycling. And then, because we will have developed drugs for each of those pathways, pair the genetic mutations with the pathway, with the drug, to make sure that the right ALS patient gets the right drug at the right time. That's the future. And now to kind of talk about the nitty-gritty of the present is Elizabeth Harrington, our genetic counselor at Columbia University. Thank you.
Okay, great. Thank you, Dr. Harms. Let me quickly share my screen. So I'll just mention my quick disclosure here. And, oh, my clicker's not working. Let's see. There we go. So I'm going to be talking a bit about how specific genes are leading to clinical drug trials. So first I'll talk about specific gene and gene-related therapies, where they are in clinical trials, and then some other observational studies. And then towards the end, I'll talk about considerations for genetic testing, implications of results, kind of how one goes about genetic testing and who should get testing, and how this really relates to those who are listening. So first I'll talk towards antisense oligonucleotide therapies, or ASOs. And Dr. Harms did a great job of describing the human genome and the importance of proper protein functioning. So again, this is a diagram of the central dogma and how proteins are made from DNA. So to begin, we have DNA, and that's transcribed into what we call messenger RNA, or mRNA, which we can think of kind of like an instruction file. And the mRNA carries the information from the gene to a compartment of the cell that builds proteins, and then the information is used to convert it into a protein. So we can use the gene SOD1 as an example that Dr. Harms mentioned. And this provides instructions for making an enzyme called superoxide dimutase. And that's abundant in all cells throughout the body. And it typically binds to molecules to break down toxic molecules. When we have a mutation in the SOD1 gene, this can lead to destabilization and oxidative damage, and contributes to that misfolding and clumpy aggregation that Dr. Harms was speaking about. So antisense therapies really change the process of producing a protein kind of before it begins. So these are short, single-stranded pieces of DNA that match the complementary sequence of the mRNA. And they're really designed to seek out, to bind to, and destroy mRNA in a really specific manner so that we don't produce the amount of disease-causing protein. This is a diagram that explains what many of the ALS ASO therapies are doing. ASO therapies can also be designed to target some of those loss of function type of mutations as well by trying to restore the normal protein levels in the cell. So there are several different diseases and genes that have been developed, or ASOs that have been developed for several different diseases and specific genes. For the ALS-related genes, these are the those that are currently in clinical trial are the C9orf72 gene, the most common associated gene with ALS, the SOD1 gene, the first gene that was discovered, as Dr. Harms mentioned, and a gene called FUS, which can cause both juvenile onset ALS and a little bit more of an earlier adult onset ALS. So these are the ASO therapies, and I'll get into the clinical trials for these in a moment. But first, let's talk about some other therapies separate from ASO. So we'll talk about a drug called metformin, which is thought to being used to target and treat C9 ALS. So first about the C9orf72 mutation. This is a mutation that's a little different than some of the more common genetic mutations that cause disease. The specific mutation that's in the C9 gene that causes ALS is what we call a repeat expansion. So that just means at the very beginning of the gene here, there is a sequence that's spelled GGGGCC, and it typically just repeats itself a few times. The mutation is when it's been abnormally expanded, so hundreds or thousands of repeats. One of the consequences of this abnormal repeat expansion is that it unexpectedly produces these rogue proteins called RAN proteins. And these RAN proteins can accumulate and be associated with causing disease. So metformin is a widely used drug to treat type 2 diabetes. And it's also thought to be potentially a viable therapeutic for C9 ALS to target those abnormal RAN proteins. So after it was shown that metformin can reduce those RAN proteins and improve disease in C9 ALS mice, it has moved into a single-site clinical trial to see if the drug is safe and effective for C9 ALS patients. So I'll mention kind of where these different gene-specific therapies are in clinical trials, but first want to speak a little bit about what's involved in these different clinical trial phases. So clinical trials are only done after preclinical findings suggest that a new drug treatment is likely to be safe and work in a subset of people with disease. So before a drug is given in humans, it first needs to be developed, of course, and determine a potential effectiveness. It's often tested in the laboratory with different cell studies and animal models to look at initial safety data. And then, of course, there's a lot that goes into the application for approval to get into a drug trial and have the drug be given to humans. Once we get into the clinical trial, this is conducted in phases that build on one another, and each phase is designed to answer a certain question or certain questions. So phase one is really focused around if the treatment is safe. It's usually given to a smaller amount of people. In the first few, there's a very low dose given. And if there's only minor side effects, then the next participants might get a higher dose and a higher dose as we go on in the clinical trial. Within each phase, there also might be different cohorts of individuals enrolled. So it's not just one group of individuals enrolled at one time and can sometimes be a longer phase drawn out with many different enrollment points. So if the treatment is found to be safe in phase one, it moves on to phase two of the clinical trial to see if it works, to see if there's any benefit. So this is really about testing the efficacy of the drug and further evaluate the safety. So phase three really focuses on confirming the effectiveness of the drug and collecting additional information on a larger number of individuals, and then more specifically monitoring any side effects. Upon completion of the clinical trial, with enough evidence to say that it's safe and effective or more effective than any other previous drugs that have been out there, it is submitted to the FDA for approval. If they feel like it's safe and effective, it might be approved. And if there's not enough data, then the FDA might require additional studies to be performed. So where are we in the specific clinical trials for specific genes related to ALS? So there's been a great deal of progress in the development of ASO therapies for some of the genetic forms. So SOD1, the first discovered gene, is now towards the final stages of phase 3 in clinical trial. And that drug has been named tofersen, and the trial name is called the Baylor trial. The C9orf72 ASO is towards the end of the first phase of clinical trial. And this should be moved over to phase 3, actually. There's also been a ASO developed for the FUS gene, which again causes the earlier onset disease. This was first given to one of our patients at Columbia and then extended to an expanded access program, allowing several other individuals with FUS ALS to receive the therapy, and is now advancing into phase 3 of the clinical trial, which will hopefully open up at multiple sites around the US and internationally. Metformin is in phase 2 of clinical trial. Because this drug was used and is safe for the treatment of individuals with type 2 diabetes, we are able to skip the phase 1 safety phase to move straight to seeing if it is effective to treat individuals with C9 ALS. And then lastly, with so much positive data from the SOD1 ASO study in those who have ALS, it was recently announced that there will be a presymptomatic clinical trial for those that carry SOD1 mutations but do not yet have disease. And that clinical trial has been named ATLAS. So how did we get here, where a drug has been developed and it's possible to test in humans and determine effectiveness? So much of that data has been used, much of the data that has been used to develop these therapies and make for a positive experience in clinical trials is from what we've learned from individuals who have these specific genetic forms of disease. And these are often called observational or natural history studies. One of the goals in these studies is to identify what we call biomarkers, which is just a measurable substance whose presence is indicative of disease. So these usually correlate with disease. Sometimes they can be a predictor of the onset of disease or progression of disease. And since it's measurable, it can often be used to determine if a drug is effective or not. So when there's a drug given, do these biomarkers change or stay the same? So much of what led to the C9 and SOD1 clinical trials is from the data learned from individuals who participated in these C9 and SOD1 natural history studies. And similarly, Dr. Harms is leading a FUS natural history study to gather more insight into the individuals with that particular subset of disease. So yes, our goal is to have an effective treatment for those with disease, but specifically for individuals with genetic forms of disease, we can also aim to develop therapies that prevent disease from occurring. So that's really the goal of that SOD1 presymptomatic ASO trial. And the question is, how did we get there? Or really, once that trial begins, when we're treating individuals who don't have symptoms, how will we be able to determine if the drug is effective if they don't have disease symptoms? Well, one of those items is those biomarkers that we mentioned. And those are really gathered from natural history studies of individuals who don't have disease symptoms, but are carriers of these genetic mutations. So these types of natural history studies are available and for individuals who are carriers of these genetic mutations, and it's invaluable to learn more about the earliest signs and symptoms of disease. So at Columbia, we have designed a study called the ALS Families Project. It is targeted at individuals and families who have genetic forms of ALS, but those individuals who are asymptomatic. So the first study provides predictive genetic testing and counseling to those who wish to learn their personal genetic status. And the second is more towards the natural history study. So seeing individuals over time, about once a year, collecting different biospecimen samples and clinical data to learn more about what's happening on a cellular level before the onset of symptoms. So again, this is for individuals who have an affected family member who has a genetic type of ALS. So going back to how this is all relevant to you is really the first step in all of this is genetic testing. So there's many different things to consider when thinking about genetic testing, and I'll list some of the reasons why individuals choose to pursue genetic testing or may choose to not proceed with testing. So some individuals, it might help determine the underlying cause of disease. This is mainly the case when we have an abnormal mutation that's come about on testing. And that can help confirm a diagnosis and help us understand what's really causing disease. Some people are just curious about that. It may inform you about potential clinical trial opportunities. So this talk is really focused on the genetic forms of disease and clinical trials around those. There's also other clinical trials that are not specific to genetic forms, but learning your genetic results can help us focus on what clinical trials will be most appropriate and potentially effective for you. It can help provide further knowledge and information about risks to family members. So if you yourself, an individual with ALS, is tested and found to have a specific mutation, we know that most of these genes are what we call autosomal dominant in nature, meaning you have a mutation in one of your two copies of that gene, and you pass half of your information down to your children, share half of your genetic information with your siblings and your parents. So first degree relatives are at a 50% chance or risk of having that same gene mutation. And some people are interested to learn that risk for family members. It can help them plan for their future. And oftentimes people will have to do testing to either make family planning decisions for themselves or help their children make those decisions. So there's now the genetic technologies to use a process called in vitro fertilization and screening embryos. If there's a genetic mutation that's known in the family, there's additional reproductive options that might be taken into account for some individuals. So on the other side of things, some individuals choose not to pursue testing because it might not change your care plan. There's no FDA-approved specific drugs available to treat these genetic forms right now. Or a lot of these are just in the clinical trial stages, as we mentioned. So if you're not interested in these clinical trials, and that would have been a main motivator, some choose to not pursue with testing. There can also be a risk to some of these clinical trials, and so individuals are dissuaded from testing, again, if that's one of their considerations. And of course, there's the other side of learning the risk for how this information might affect your family members. And we know that there is a lot going on in the family as it is right now, and the burden of learning this information might not be beneficial. And so it's completely reasonable to choose not to learn this information for that piece as well. The cost of testing used to be a bigger factor. It's less so now. There's a bit more, there's some sponsored testing programs that have brought down the cost of testing. And generally the cost of testing used to be thousands of dollars and now is in the hundreds. So this is I think, less a factor now than it really used to be. And lastly, there are some potential insurability discrimination risks that we do bring up. This is less so related to you. Essentially, if you, an individual with disease, get tested and are found to have a disease-causing mutation, then before sharing that with your family, or if they themselves would want to get tested, we would make sure they are properly insured prior to undergoing testing themselves. So how would you get tested and really what's involved? Speak with your neurologist, your ALS specialist, a genetic counselor. They'll have you sign an informed consent. It's typically done by a blood draw or a saliva collection kit that can be sent to your home. That is sent to a commercial genetic testing laboratory, and we usually get results in a few weeks. So what results can be returned on this testing? So a negative test result. Again, we're really focusing on testing the individual with disease. So if that person does not have any known gene mutations and no known family history, it doesn't mean, this negative result doesn't mean that you don't have this ALS diagnosis. It just means that we've not identified a genetic cause of disease. You can also get a positive or abnormal result, meaning a disease-causing variant was identified and can confirm a diagnosis. We talk about the implications for you, clinical trial options, and family communication of this result. There's also a third type of result called a variant of uncertain significance, or a VUS. And that just means that upon doing genetic testing, reading through these genes, a change or a variant in one of these genes was identified, but we don't have enough information to tell, is that a harmful gene that leads to the protein clumping that Dr. Harms recommended, or not enough protein? Or is that just normal variation and doesn't actually change the function of the gene? So I have this little cartoon here. You can picture that that's Dr. Harms reading through everyone's research genetic testing, and a big part of genetic testing is the interpretation of these variants. And so ClinGen is a clinical genome resource. Dr. Harms is leading kind of the charge on the ALS gene curation expert panel. So improving the interpretation of these genomic variants and genes associated with ALS. And that will really further our clarification and results that we are able to give back to those who are getting testing themselves. So just some last takeaways, and we'll open it up for questions. Single genetic causes for ALS are really most commonly identified in individuals with a strong family history. So if there's no strong family history of disease, it's less likely that there's a genetic cause of disease. It's not zero, but that goes back to those kind of pie charts that Dr. Harms was showing. The actual disease risks for family members really depends on that penetrance that he was mentioning. So one having a specific gene mutation doesn't necessarily mean that 100% you will develop disease symptoms yourself. We are doing genetic testing more as these clinical trials are becoming more abundant. And it's important to recognize all that goes into genetic testing, the potential benefits and drawbacks, and all can be considered and talked about with your specific clinician when thinking about doing this for yourself. So I'll end here, and I'm happy to, I think we have some time for questions now with Dr. Harms and myself.
Yes, we do. We have some excellent questions that have come in. One is in the case, and I think Dr. Harms, this is for you, but I'm sure Ellie as well. In the case of spontaneous mutations, do they occur in utero? And then sort of a follow-up question to that is, if sporadic mutations don't occur in early embryo, when do they occur?
Right. So there are two places where an apparently spontaneous mutation could take place. It could actually be in either the testicles or the ovary. And so there could be a small number of sperm or a small number of eggs that have the mutation that then go on to become a baby. If that's the case, then there's a small chance that that same pocket with the mutation from the eggs or the small pocket from the sperm would go on to create another affected individual. We don't know of any cases like that right now in ALS. The more likely thing, and we know this from other diseases, is that the most likely thing is that like one sperm or one egg develops the mutation. That's probably 93% of the time. But again, because most people develop ALS at a time when their parents are already deceased, it's unusual for us to be able to look and see if a spontaneous mutation is at play, unless it's one of those very early onset cases. And then we usually find just one mutation, and that's a mutation in the FUS gene. So those are the situations where spontaneous mutations, de novo mutations is their technical term, have been found.
Great, thank you. Next question is there value in genetic testing for family members who may have inherited C9 before they exhibit symptoms of ALS? And at what age is testing appropriate? If the gene is detected, are there any prophylactic measures that can be taken to prevent or delay ALS?
Ellie, you want to take that one?
Yeah, I'll take that one, and maybe you can speak more towards the last part of the question on prophylactic measures. So I think, it's a very good question, and there's a lot to consider in pursuing predictive genetic testing. It's a very personal decision and not one that's necessarily recommended across the board. But I would say if it's a question, what's recommended is to have genetic counseling to really talk through those potential benefits and drawbacks for you specifically, as many of the implications might be more psychological in nature. We want to make sure that you as a person have specific motivations and really want to learn these specific results and know the implications of them. So I don't think there's a right or wrong answer here. I think it's all on a very individual basis. And we're happy to that's one of the main jobs of a genetic counselor is really walk you through that process of helping you come to a decision that's right for you right now.
And I'll add that at the moment, the main way that learning your status before a drug is widely available is the ability to make fertility decisions around not passing the gene on. There may come a day soon, if the C9 antisense oligonucleotide, or if it looks like metformin is proving to be successful in C9 carriers, then there may be prophylactic steps to take, but we're not there yet. And so as soon as prophylactic steps are available and are scientifically proven, that will change the algebra about who gets testing and who doesn't in a presymptomatic state, and definitely bias people towards getting tested so that they could take advantage of potential prophylactic medications. But at the moment, those don't exist.
Thank you. We have another question a little bit similar to your answer, but slightly different. Is it recommended for family members to get genetic testing if one parent, such as 87, passed away, who basically lived like Superman, except until the last 10 months of his life?
Yeah, so, the penetrance is a funny thing. One of the things that I didn't have time to say is that we've estimated statistically that on average, an individual has to have 6 of those insults in order to develop ALS. Now, some genetic mutations represent all 6, like it's a bad enough insult that it's basically like having 6 small insults, right? And so most mutations, though, are in the category where you need at least one other insult, and we don't know how to identify that yet. And so it's entirely possible, though unusual, for someone to live to 87 and not experience a second hit, but it is within the realm of possibility. So it doesn't exclude you from considering genetic testing. It would still be wise to think about your personal philosophy about that and to have a discussion. But there are people who have lived to 87 with the C9orf72 mutation and never developed symptoms before their death. So it does happen. Ellie, anything to add there?
I'm just taking a peek at the question. I think the question was a little bit more towards if for family members of someone who's 87 with ALS. So, if there's no other family history, the likelihood that that's a genetic condition, I would say, is low, especially given the age of onset of disease. And so unless we do have a strong family history of disease, we typically do not offer testing to family members of someone without an already established gene mutation. So the first line of testing is really always the person with disease. Only if something is identified on testing, then we say, okay, other family members can, we would test specifically for the known gene that was identified on testing.
Yeah, thanks for catching that. That is really important, a very important point.
Thanks, Ellie. We have one more question. Are there findings from clinical trials relating to genetic ALS helpful at all in understanding and potentially finding treatment for sporadic ALS?
Yeah, absolutely. So, most of those pathways that I was illustrating, we've learned about from the genetic forms of the disease. And then you go and you look in people who don't have an identified genetic problem as of yet, and you see those same pathways being disrupted. So from the C9orf72 patients, we learned that the transport of RNA from inside the nucleus to outside the nucleus is dysfunctional. And then you go and you look in sporadic patients who don't have an identifiable gene mutation, and you see that same problem is present. So now you can say, well, okay, we should try to target that shuttling mechanism because we see it in some of the genetic forms, but we also see it in the the gene negative cases at this point. So yes, we're absolutely learning a ton about sporadic non-gene negative disease that we're going to be applying in that case.
Great, and we have one follow-up question to the C9. Does C9 usually need additional insults in order to trigger ALS symptoms?
Yeah, so the average, so here's one bad thing, and that is that growing old probably counts as 2 insults. So yes, C9 needs another insult, but for most people, for probably 70% of people, getting older is enough of a second insult to trigger disease. So there may be others that we haven't discovered yet, but it is true that that there are other insults needed, but unfortunately age, which none of us can avoid, is a pretty strong insult.
Thank you, Matt, and thank you, Ellie. Unless anyone else has other questions, we'll move to wrap. But if you may have other questions that you think of after, feel free to let us know and we can certainly help facilitate those.
Thank you both so much for participating in the session today. We really appreciate all the answers and the very informative presentation that you both gave. Thank you so much.
Thanks for having us.